Asexual reproduction does not involve the union of gametes or fertilization, and therefore only one parent produces offspring that are genetically identical to the parent. Now, this can be very beneficial in a stable environment because it's not going to matter that they're genetically identical, and it's going to cost very little energy to make that offspring. But in an unstable environment, that can lead to major problems. Now, there's a wide variety of asexual reproduction. Binary fission is asexual reproduction in prokaryotes, very similar to mitosis, which is asexual reproduction in single-celled eukaryotes, which makes sense because mitosis evolved from binary fission. But when you go into multicellular organisms, they get a little more complicated. You have budding, like you would see in a hydra, where the organism buds off, spores, like in a fungus, fragmentation, like in a planarian worm or in a sea star, where you get cut and they regenerate the organism. Vegetative propagation is when plants bud off, like those spider plants where you can plant the little spider plant or so on. Parthenogenesis is a weird one because that is when you have females only for reproduction. You don't actually need the males, so that can lead to some interesting situations. So the example in the video here is that when a queen bee wants to make a drone bee, they don't need a male. But if they want to make another queen or a worker bee, then they do. Populations of sexually reproducing species generally have much greater genetic diversity than do populations of asexually reproducing species. So sexual reproduction requires fertilization, or the fusion of gametes, and uses the process of meiosis to create haploid gametes. Haploid means having half the number of chromosomes. If we look at the example here, humans have 46 chromosomes. That is our diploid number. But at the end of meiosis, we produce haploid gametes, or ones with half, or 23. When an egg and a sperm come together to make a new organism, or a zygote, you do fertilization to create 46 again. So offspring produced by sexual reproduction are very different from the parents because of new gene combinations resulting from a variety of sources that we're going to talk about in this video. So the process of meiosis is essential to sexual reproduction, just as mitosis is to asexual reproduction. Sexual reproduction, like we said, requires fusion. And if I had gametes that were diploid, I would end up with an organism with way more chromosomes than is actually sustainable. Now, when you actually do these, both chromosomes or both gametes have matching chromosomes called homologous chromosomes. So it's two chromosomes containing the same types of genes. One chromosome from the pair is from the male, one chromosome from the pair is from the female. And these chromosomes are going to match up and create the organism that you are familiar with. Now, meiosis did evolve from mitosis, so the stages are going to be very similar, but you need to be aware that the processes are radically different. If you look at the standard human karyotype, you can see that we have 46 chromosomes. Now, if you were to go through and align these, you would find out that we have 23 pairs of homologous chromosomes. So this is the aligned picture in a human female. So you can see that all chromosomes have a matching pair. Now, this is only going to be true of females in humans because males don't have two X chromosomes. Males have an X and a Y. So if you look at a male, you can see that that's not always going to be the same. But we still have two sex chromosomes, and we have two of all of the other chromosomes. So we are diploid organisms, or 2N. Unlike mitosis, meiosis has two steps. In meiosis 1, the homologous chromosomes separate. But in meiosis 2, the sister chromatids separate. Now, where do we actually become haploid? We become haploid at the end of meiosis 1, so they stay haploid. But if you look at the chromosomes, in meiosis 1, the chromosomes are still double-stranded. They have two strands. But in meiosis 2, they are single-stranded, and that is a major difference. Interphase in meiosis is just like interphase in mitosis, but it only happens once. It only happens prior to meiosis 1. Chromosomes still duplicate in the S phase just like normal, but like we say again, it only happens once. If interphase happened before meiosis 2, then meiosis would be just like mitosis. Everything would be copying again and again, and I'd have 2N making 2Ns. But meiosis is reductive. You go from 2N to N. Prophase 1 is very different from prophase in mitosis because instead of the chromosomes all lining up on the metaphase plate, the homologous chromosomes pair up. Now, because they are pairing up, they're connected by a process called synapsis during prophase 1. That means that they are going to be very close to each other, and an exchange of genetic material can occur. These overlapping sections happen called chiasma, and the chromosomes are fairly fragile, and they break, and the pieces exchange, as we can see down here, making a very different chromosome than what they had before. So this is going to be one of our major sources of genetic variation in meiosis. In metaphase 1, the paired homologous chromosomes are aligned along the equator of the cell with one chromosome of a pair on one side, say the mother's one, and one chromosome of a pair on the other, say your father's. Each pair is randomly oriented in terms of whether the paternal or the maternal chromosome is on the given side of the equator. This arrangement is called independent assortment and causes daughter cells to have DNA that is different from the original parent cell. So independent assortment, I should say, is one of the major sources of variation in sexual reproduction. In anaphase 1, the homologous chromosome pairs are going to separate and move to opposite poles of the cell. This isn't like anaphase in mitosis where the sister chromatids separate. This is the homologous pairs separating from the tetrad. So each daughter cell will receive only one chromosome from each homologous pair. Now, the actual movement process occurs exactly the same way as it does in mitosis with motor proteins and kinetochores and so on. In telophase 1, we see the reverse of prophase, just like we saw in mitosis, but you'll notice that the chromosomes, like we said, have sister chromatids. So at this point, these cells are definitely haploid. So each daughter cell is going to proceed from meiosis 1 and go into meiosis 2. Some cells do cytokinesis here, some don't. It's going to vary depending on the organism. If cytokinesis occurs after telophase 1, it technically is called something different. It's called interkinesis. When you look at meiosis 2, the steps are pretty similar to what you would see in mitosis. The problem here is that it's happening twice. So the end result, without having an interphase here, is that I'm creating 4 haploid daughter cells. So that's going to be a bit different, but overall the process is very similar. Now, where do you actually use meiosis? Well, you use it in the production of sperm and eggs. If you are producing a sperm, then you are going to be going through the full process of meiosis and producing 4 spermatozoa, which you would normally think of as sperm. And males do this all the time, producing many, many viable sperm every day. But if you want to produce an egg, it's going to work very differently. If you'll notice in the diagram, only one full ovum is produced for each meiosis event. What happens is that an egg requires a lot of cellular machinery to be able to divide fast in the first couple of days of the reproductive process. So it takes all of the cellular machinery that would have gone into 4 eggs and puts it into 1 with the DNA of 1. The leftover DNA is discarded as these polar bodies off to the side. So spermatogenesis produces 4 haploid sperm, but oogenesis only produces 1. Now, oogenesis is a much more complicated process, and it occurs prior to birth. So every egg that a woman will ever have has been created prior to them being born, but they're frozen in prophase 1, waiting for puberty, and then once fertilized, they will eventually continue along the process of meiosis. Why is meiosis important? Well, meiosis is important because it introduces genetic variation. We talked about random assortment in metaphase 1. If you really think about it, that means that there's 2 to the 23rd different combinations where the paternal and maternal homologous chromosome that you have would be aligned in your gamete. So that means that there's more than 8 million combinations just within you. If you then look at which gamete from you and which gamete from your partner come together to make that zygote, that means that there's around 70 trillion different combinations between any 2 people. And if you expand that even further and look at prophase 1 and crossing over and that randomness that occurs during synapsis, well, the actual differences here are functionally infinite. And so this is where the variation that comes from in sexual reproduction, and this is essential to our evolution and our survival in an unstable world. You need to be familiar with the differences between meiosis and mitosis. There are some major differences, so look over this diagram as you have time and make sure you can understand it. If you have any questions, come talk to me.